All right, so we're going to pick up now and take a look at some of the kind of early attempts to figure out what exactly goes on with plant development during the production of the seed itself. So the way that they did this was they actually looked at mutant strains of what are called Arabidopsis plants. Or Arabidopsis is something in the mustard family. It's used because it grows pretty quickly, and you can follow it through its life cycle and track its genetics relatively quickly. You don't have to wait for decades to have this thing grow and produce seeds and reproduce, for example. So what they found here by doing this experiment, looking at these mutant strains, was that if you have a plant, for example, that has altered development in the embryo, aberrant just means it's altered, it's not normal, it's different in some way. What actually occurs here, if the development of the embryo is off, then what's going to occur is the suspensor cells are going to start to grow like an embryo, which isn't going to work real well, but it is kind of like the built-in way the plants have to try to correct or account for or get around the issues that might be going on with development. If you have an embryo that forms normally, those suspensor cells develop like they're supposed to into a suspensor. They don't develop, for example, into an embryo. So again, the situation here is you want, as a plant, to have both the embryo, which is going to grow into the plant itself, as well as the suspensor, which turns into the other cells that we talked about previously. We see over here, this picture here, normally, if the embryo forms the way it's supposed to, it's going to prevent the suspensor from forming into an embryo. If, on the other hand, the embryo doesn't form properly, then these cells down here are going to try to become an embryo themselves, because that's really the critical part of the plant itself. All right, so what happens here, once the embryo and suspensor have started to form, once they start to grow and develop and go through cell division and so forth, the relative position of the plant is established very, very early on. So for example, we talked before about the root-shoot axis. That's formed really, really early on. That vertical axis where the plant is going to grow and develop happens relatively quickly. What also happens here is the radial axis. So the part of the plant sort of going around the stem, circular for example, is the radial axis. That also develops relatively quickly and is set up pretty early on in the development process. In this embryo, the solid ball of cells, what eventually starts to happen pretty quickly is, depending on where a given cell is located inside this solid ball of cells, that will determine what kind of cell that cell will become. Okay, so for example, if we have cells located relatively far to the outside of that solid ball of cells, those cells are going to become dermal cells. Relatively, later on, they're going to become dermal cells. If they're located somewhere in like the middle part of this, right, so in here, or maybe even in here, you're going to have cells that become ground tissue cells. And if you have cells that form kind of in the midst of this, more or less what this ring is in this picture, those cells are going to become vascular tissue cells. Okay, so we have all these different types of cells really, really early on developing, still inside the embryo. This would still be actually inside of the seed. This is not germinated yet. It's not started to grow yet. It's still located inside the seed itself. And so these tissues are located around this root-shoot axis radially, or in a circle around the root-shoot axis. That's where they're going to be located. And so eventually, over time, as this root-shoot axis is going to elongate and develop and grow and get more advanced, then we start to see the beginnings of the shoot apical meristem up here. We've got what will eventually become the cotyledons later on in life. And then again, we have vascular, ground, and dermal tissue as well being shown relatively quickly and early on. All right, so actually a micrograph now of what's going on here. We have the embryo, the more or less what becomes solid ball of cells, and then the suspensor down here. So as time goes on, the cells divide, there are more cells, but the relative position of things hasn't really changed very much. We advance a little bit further, and now we have something which begins to look a little bit more plant-like. We're starting to just get a sense of where those cotyledons are going to show up over here. So they're just barely kind of popping up, and if we fast forward now a few more intervals of time, we're going to actually now see cotyledons. We could actually see ground meristem and protoderm on the outside and procambium all the way in the core, which of course we know becomes vascular tissue. So these are the primary tissue types, which then give rise to the actual functional tissue types later on in life. All right, so as it turns out, if we sort of fast forward again a bit more into the development process, what we find out is that the roots and shoots are controlled independently of each other. All right, so again, the way they've found this is by looking at mutant strains of these different plants. There is a gene in the Arabidopsis genome called shoot meristemless, or just the STM gene, which you have to have if the shoot's going to form normally. If a plant has a mutant form of the STM gene, or if it's missing the STM gene altogether, then what's going to happen is these plants don't really make a shoot. Right? They're not going to have the right protein. It's going to allow them to produce the proper above ground part of the plant. So they're going to basically be the cotyledons, and then a bunch of roots. The root system looks perfectly normal because their genes are perfectly normal, but there is no shoot or above ground part. Now, why is this important? Right? These plants clearly then are not going to survive. They're going to fail. They're going to die. They're not going to make it because they don't have an above ground part. They can't produce the above ground part, so they're not going to make it. But what they do is they show us how development works. What this has shown us is that the roots and shoots grow independently of each other, starting off from different genes from each other. And so they're very much an independent pair of organ systems in these plants. Again, these STM mutant plants do produce roots, but there is no stem produced at all by them. All right, so there's another one here called the Hobbit gene. All right, some of these genes have some fairly creative names. This Hobbit gene is one kind of like STM, but this one now is for roots. And so if a plant does not have the correct form of the Hobbit gene, then they're still going to produce the above ground part because those genes, STM for example, is perfectly normal and functional. But if they don't have the correct form of the Hobbit gene, they're not going to produce root meristems. And if they don't have root meristems, then obviously they can't produce roots either. All right, so now in this case we have the protein. This protein that allows for the roots to grow is one that actually allows auxin. This is a very, very important plant hormone. Auxin is one of the more important ones. And auxin is one that actually turns on the gene or genes that you need to make the root meristem. Right? So Hobbit is a gene that makes a protein. This protein lets auxin turn on other genes to allow for the production of roots. So it's kind of like a cascading series of steps where eventually you get to have roots. You've got protein produced by Hobbit. We have the protein allows for auxin to do its thing. Auxin then allows these other genes to do their thing. And in the end, what you get is the production of healthy roots. All right, so this auxin hormone, like I mentioned, is a very, very important one. What's involved here actually is the activation of what's called a transcription factor. Okay, so what happens here is that we have another gene called monopteros, or just MP. This is a gene that allows for the production of an auxin-induced transcription factor. Auxin-induced transcription factor. There's a lot going on here on this slide. Basically, what's happening here, because we haven't covered the chapters on gene expression and transcription and translation and those kinds of things. Basically, what transcription is where you take DNA and you turn that into RNA. Right? So down here on this picture, we have a chunk of DNA. This gene is a root development gene. This is DNA. If we transcribe it, we get RNA, mRNA in this case. And mRNA can then be turned into proteins, which allow roots to develop. Okay, but just kind of in a nutshell here, a very, very Cliff Notes version, almost like an outline form of what transcription and translation do. In order to have transcription, there has to be what's called a transcription factor. Transcription factors are things that bind to what's called a promoter on a gene and allow for transcription to begin. Okay, if you don't have this transcription factor, you cannot do transcription, you cannot make RNA. If you can't make RNA, you can't make protein, and if you don't make this protein, then you don't get roots. Okay, so what's happening here is we've got this monopteros structure up here, which has a repressor on it. The repressor prevents monopteros from doing its job, but if you take away this repressor by having auxin present, so auxin, what it does is it allows for this blocker to be removed, which then allows for monopteros protein to go ahead and be the transcription factor for this particular gene. So again, there's a lot to this, right? We've got a transcription factor that currently is blocked. If auxin is present, you take away the blocker. And if you take away the blocker, monopteros protein can then bind to the promoter, allow for transcription, allow for the production of a piece of mRNA, and once you have the mRNA, this can be translated to produce a protein, and this protein actually allows you to go ahead and make the roots, form that root apical meristem, and go ahead and get started. All right, so basically what this is saying is what I just went through, and I would highly encourage you that if you didn't follow that, go back and listen to that little section again, because we didn't cover transcription and translation, and you might be completely lost. If you haven't had an Intro 1 course, if you haven't had the cell biology course that talks about how transcription, how translation work, you're maybe, feeling like I'm talking in Greek right now. But if you go back and listen to that again, hopefully it will be a little bit clearer. Okay, so we've got this protein, Monopteros. This green protein is produced by the Monopteros gene. But normally it comes set up with this little orange molecule here called a repressor. The only way to get this repressor off is to have auxin, which is a plant hormone. It's produced elsewhere in the plant. Auxin will allow this blocker to come off and allow the Monopteros protein to do its job as a transcription factor at the promoter. Once this green oval binds to the promoter, then we can go ahead and have transcription of the root development gene. This is a piece of DNA. Once that's transcribed, we get a piece of RNA. Once that's translated, we get a protein. Once we have that protein, we can go ahead and do our formation of roots. Okay, so again, here's kind of a look at really early, super young embryonic stages of plants that have either the Hobbit or the Monopteros. Remember, both of these are involved in root formation, and you really have to have both of them in order for roots to form normally. This is what the plant looks like normally. And again, we're talking really young embryonic versions of plants here. So it doesn't really look too much like a plant yet, but it would if it were allowed to grow. So here's one where both Hobbit and Monopteros are the wild type, and wild type just means normal form of both of those genes. Here's one where we have the Hobbit wild type, but the Monopteros is a mutant strain. So now we've got cotyledons, there'd be a shoot forming here, but the roots didn't really do very much. There's not a whole lot of branching taking place down here at the roots. That's not going to be good. Up here we have the Hobbit mutants, but Monopteros normal, and what you see here is, again, the roots don't look normal. They don't really long, kind of primary looking root here, but the branching that's supposed to occur hasn't, because now the Hobbit form of the gene is also not functional. So neither one of these two strains would be viable. Neither one of these two is going to live for very long. You might say, well, why are we bothering talking about this? Well, this is how we learn, right? By looking at strains of plants or animals that are abnormal, that allows us to see how the normal versions get to be normal, how they express genes they have to express, how they grow the way they need to grow in order to take on that normal form. Okay, so now the tissue systems. We talked before about our primary meristems. Again, primary meristems are your ground meristem, your procambium, and your protoderm. Those are developing even when the plant is still in that solid ball of cells, kind of globular stage. Those primary meristems are forming. So it's not like in an animal where you have cells migrating during development. One of the things you see in an animal as it develops embryologically is cells actually move around. They migrate within the embryo to get to places where they need to go to take on the form of tissues they need to take on. That doesn't happen in plants. In plants, wherever a cell is set up, wherever it starts off physically, wherever it is in terms of its location, that will determine what kind of cell it will eventually become. That kind of makes sense. The physical position of the cell determines what kind of cell that cell is going to be. So again, we have the protoderm, which is one of our three primary meristems. Protoderm becomes dermal tissue. We've talked about that before. We know dermal tissue is the outer covering of the plant itself. The ground meristem becomes the ground tissue. Again, we've seen this before. The ground tissue is kind of like the foot soldier part of the plant. Most of the body of the plant is made up of ground tissue, and that originates from the ground meristem. And then the inner part is the procambium, and that's going to give rise eventually to the vascular tissue. And vascular tissue, as we know, is involved with transporting things like waters and sugars and so forth throughout the body of the plant. So these are our three primary meristems that are produced by the apical meristem that eventually give rise to our different tissue types. Again, our globular stage, and when you think globular, you're thinking solid ball of cells all the way back, super early in development. This is also where the cotyledons are produced. Cotyledons, remember, are the seed leaves. They're the very first leaves this plant will ever have once it gets out of its seed. They, in dicots, dicot actually means two cotyledons. Monocot means one cotyledon. And these cotyledons are produced differently from all the other leaves that plant will ever have, right? Those early primary seed leaves that a plant produces are not the result of the apical meristem. They're the result of embryonic cell production. So from that point on, after the cotyledons are in place, every other leaf, thousands and thousands of leaves, if you're looking at a big, long-lived tree, maybe hundreds of thousands of leaves, they will all be produced by apical meristems, whereas these early first seed leaves that are produced from the embryonic cells. Very, very different kind of origin for those leaves. And so that process of producing cotyledons is called morphogenesis. The beginning of the form of the leaves is called morphogenesis. And in the case of the embryonic production of these cotyledons, it's the result of differential growth and differential directions that result in production of the cotyledons themselves. From there on, it's positioning of the cell plate. And again, we didn't cover mitosis in Biology 1020. So a cell plate is something that happens during cell division. And at some point during cell division, there has to be a point where you take one cell and you physically divide it in half. Right? So picture a cell like an oval, and you could either have a division happening from top to bottom, or you could have a division happening from left to right to produce two cells. Depending on where the cell plate is, the cell plate's like the divider that splits a cell in two. Depending on where the cell plate is and depending on the direction it's going, that's going to determine which direction the cells are dividing in. Okay, so we'll take a look at that as well. There's also a function here of part of the cytoskeleton, microtubules and actin filaments that also are involved here in allowing cells to divide in one direction or another. Cellulose also plays a role because some parts of a cell wall are thicker, and it would be in the plant's best interest to not try to divide cells in a direction where the cell wall is very, very thick. It would be very difficult to do that. So oftentimes when it comes to cell division in plant cells with mitosis and cytokinesis, what you'll find plants doing is taking the path of least resistance. They're going to divide in the direction where the cell walls are the thinnest, because that will be the easiest way to chop that cell into two cells. We'll see that here in a second too. So they're going in, like I said, the path with the least reinforcement of cellulose. So up here, two different alternative ways that a cell plate could form. Cell plates could form top to bottom. So the microtubules here are going to form the cell plate top to bottom. And what eventually happens is there will be a divider here in this cell that produces a cell on the left and a cell on the right. Another option would make a cell plate from left to right, which would mean the new cells are going to form top and bottom. So if you divide the cell this way horizontally from left to right, you're going to have a new cell on the top and then a new cell on the bottom as well. Right? The second part of this, looking at the direction where cellulose is, when it comes time for the cells to grow, right? There are lots and lots and lots of cellulose fibers wrapping around the cell, kind of like front to back, the way this is positioned when you look at it on the screen going around this way. These are like strings wrapping around the cell. These are long chains of cellulose fibers that give this cell lots and lots of reinforcement, make it nice and strong. But if you're talking about making this cell grow, for example, it's going to be really hard to grow outwards, like in the upwards and downwards and like toward you and like back here, away from you direction. So when this cell is growing, when it's filling up with water, if you were to pump water into this cell, the easiest way for it to grow would be from left to right, because there's less reinforcement here and less reinforcement over on the other side. If water is pushed into this cell, the cell is going to kind of like balloon out in the left to right direction. It's not going to get too much bigger top to bottom, but it's going to get a lot bigger left to right. So it's actually growing there in that direction. So that's when it comes to cell expansion and growth. It's the path of least resistance. Wherever the most cellulose is not the direction where growth is going to go. It's going to go, in this case, left to right. All right. So again, with morphogenesis, we talked about position of cells determining the cell fate. Morphogenesis involves production of not only cotyledons, but also production of other structures which might develop during the embryonic phases of this plant. So this is kind of just a bit of review. The meristems are in place. By the time, and again, this, just to kind of put this all in perspective, big picture here, this seed is not germinated yet. We're still talking about producing the seed, not growing as a plant yet. We're still producing a seed at this point in time. So by the time that seed goes dormant, by the time it sort of goes into its resting stage, where it might stay for decades or centuries or even thousands of years in some cases, seeds have been found in Egyptian tombs that are still viable. You can take them out and actually plant them and they will grow. They've been in there for 2,000 or 3,000 years and they're still perfectly viable or pretty viable. By the time that seed goes dormant, everything has to be in place. Everything has to be there, ready to go, on the off chance that seed actually gets to a place where it can hit the soil, get some water, break dormancy, and actually start to grow. So everything has got to be in place. The different meristems, the apical meristem and the root and the shoots, the primary meristems, the protoderm, procambium, and ground meristem, those have to be in place as well. And so what eventually happens is that seed will germinate, break dormancy, and it's going to start growing. Right? The apical meristems are going to start growing first. The roots are going to grow down, the shoots are going to go up, and at that point in time, the plant has entered into a very, very different and important part of its growth cycle. Right? So Couple of other things here. We see with the angiosperms only. Angiosperms, remember, are the flowering plants. They do not refer to the gymnosperms, like pine trees and so forth. They don't refer to ferns, and they don't refer to mosses. Those are other groups of plants. Angiosperms have some unique stuff that other plants don't have. Number one, previously we talked about double fertilization, and we said one of the things that's produced in double fertilization is the endosperm. So here we have, in a monocot especially, like corn, monocots have a huge amount of endosperm. All this tissue inside of here, this material inside of here, is basically starch, which initiates itself as the endosperm. The embryo here only takes up like half the space. The rest of the space is dedicated to food storage for that very, very young embryo. So that's a nice little boost of energy to get this thing growing early on during its growth. The second thing that angiosperms produce is a seed coat, which gives them protection, gives them survivability during dormancy. It's also a way for them to break dormancy. Some seeds will not actually start to grow until their seed coat has been removed or at least damaged. It has to be burned off or scraped off partially, or sometimes some seeds actually have to pass through the gut of a bird or a mammal before they'll grow. They actually have to be partially digested. The digestive enzymes in the gut have to remove some of the seed coat before that seed can actually start to grow. So it's a way to ensure, it's kind of like a timer really. If a seed's eaten by a bird, by the time that seed comes out the other end, chances are that bird has moved somewhere. And that seed's going to get a chance to grow out away from its parent plant, so it's not competing with them. It takes time for that seed coat to be broken down, and during that time, the bird or the animal is very likely moving away from where the seed was picked up. And again, my animations are out of order here, but talking about endosperm, it's extremely variable. Endosperm, by the way, this is something that kind of freaks people out sometimes. If you're a fan of popcorn, the endosperm in the popcorn actually is kind of the white fluffy stuff that pops out when the corn pops. That white fluffy popcorn actually is the endosperm part of the seed. Now this is not sperm. This is endosperm. This is starch, right? This is starch that the parent plant put in the seed, so that seed would actually have an energy reserve when it starts to grow. So it's basically carbohydrate. It's starch. It's no more than that. That's all it is. So a lot of the monocots, all the grains that we eat, not only corn but rice and let's see, corn, wheat is another one. Most of the grasses, actually, the part that we consume, the grains that we consume, most of that actually is endosperm. The grains are starchy, right? So that starch is in the form of endosperm produced inside those seeds. Coconuts have a different form of endosperm. They actually have a liquid and a solid endosperm. Coconut meat versus coconut water versus coconut milk. They're all different kinds of things, but they're all involved kind of in the same context of giving that seed a shot at survival at some point in time. And then the other big thing that angiosperms have is fruit. And we haven't really looked at fruits yet, but fruits are actually derived from the ovary of the flower. The ovary of the flower in an angiosperm produces the fruit. And the function of the fruit typically is going to be to attract animals that will then consume the fruits, and along with that, the seeds, because the seeds are inside the fruits. And then those animals will actually disperse those seeds. So it's a bit of a mutualistic association where the animals are getting some calories out of it, and the plants are getting their seeds dispersed. So that's a pretty cool way to go. That's something that angiosperms invented. Most gymnosperms don't do that. The vast majority don't do that. And if they do, it's in a very different kind of form. It's not a fruit, technically, even though it sometimes looks like a fruit. The other plant groups, aside from the angiosperms, don't produce fruits. All right, so we're going to pick up here in the next section, so we'll just kind of shut this one down.